Hello, everybody. Today we are talking about electrocuting cockroaches. So, I don't mean that 100% seriously, but I do mean that we will be talking about electrodes in cockroaches' brains and humans controlling them using their minds and stuff. But let's just go ahead and jump right into it. So, the title of the paper that I wanted to show you all today is called Brain-Computer Interface Controlled Cyborg, Establishing a Functional Information Transfer Pathway from Human Brain to Cockroach Brain. So I think that talking about a paper like this is really exciting for me because it starts to get at some of the fundamental concepts that go into brain-computer interface. That's something that I'm really interested in, and I want to explore more and more on this channel. But I really like that this paper goes into talking about a really simplified system, although it's not easy to simplify brain-computer interface because you're going to have optimization algorithms talked about in here, which I'm going to skim over very quickly because there's going to be some high-level math in there that it's tough for me and probably tough for y'all. But I think that it is important, though, to recognize that in science, one of the most valuable things that you can do is take a complex problem, simplify it to an ideal situation, and then explore that ideal situation to sort of get at the fundamental concepts that are at work. So without any further ado, let's go ahead and jump right into it. So let's go ahead and read through the abstract. An all-chain wireless brain-to-brain system. So the acronym that they're going to use here is BTBS for brain-to-brain system. And so whenever you see a term followed up by some acronym in parentheses, the acronym in parentheses is going to be the acronym that is referring to the word that comes right before it. So if you see that later on, just know that's why those letters are right there. So an all-chain wireless BTBS, which enabled motion control of a cyborg cockroach via a human brain, was developed in this work. Steady-state visual evoked potential (SSVEP) based brain-computer interface was used in this system for recognizing human motion intention, and optimization algorithm was proposed in SSVEP to improve online performance of the BCI. So I know I'm going through this pretty quickly, but I just want to. The abstract is just sort of get the information out there, and then as we go through the paper, we'll get into all of these things more deeply. The cyborg cockroach was developed by surgically integrating a portable microstimulator that could generate invasive electrical nerve stimulation. Through Bluetooth communication, specific electrical pulse trains could be triggered from the microstimulator by BCI commands. Again, that's brain-computer interface commands, so that's coming from a person and having some sort of recording device on them, which I'll explain later. BCI commands, and were sent through the antenna nerve to stimulate the brain of cockroach. Before I continue, I just want to say there are a few sort of grammar mistakes in this paper, which I'm surprised I'm surprised whenever I see those things in scientific papers, but they happen. Scientists are people too. But we're going to just push on through them anyways. Serial experiments were designed and conducted to test overall performance of the BTBS with 6 human subjects and 3 cockroaches. The experimental results showed that the online classification accuracy of 3-mode BCI increased from 72 point, so some numbers, blah, blah, blah. And basically they're saying that the optimization algorithm they used was helpful in increasing the accuracy of the system. Moreover, the results also show that the cyborg could be navigated by the human brain to complete walking along an S-shaped track, with the success rate of about 20%, suggesting the proposed BTBS established a feasible functional information transfer pathway from the human brain to the cockroach brain. So right there, most scientific papers that you read are not going to be that exciting in the first paragraph, but just take a second and really appreciate what it's saying. A feasible functional information transfer pathway from the human brain to the cockroach brain. That is amazing. We're sending information from a human's brain that a cockroach is then using to actually do something meaningful. And in this case, as they mentioned, the cockroach is going to be walking along an S-shaped track. So I'm going to go ahead and scroll down to some of the figures that I thought were really important. This is a cool one, but we'll come back to that. I just want to show you a little bit of what the actual experimental setup looks like. So here, let's see. Here you can see, let's make that full screen. So here on in part D of, I think this is figure 3 or 4, you can see the actual S-shaped track that the cockroach is going to be navigating. So I think that is, first of all, a really cool task to actually get at all. Like, you can sort of imagine in the future, and again, there's ethics and stuff involved, but you could imagine something like Mario Kart, where instead of driving cars in a video game, you're driving tiny insects along a tiny S-shaped track, or even cooler tracks and stuff like that. But this is obviously very preliminary to all that. But and that's probably not what these scientists are thinking of getting at. But the fact that you can wear some sort of device on your head and then you can send signals to the brain of a cockroach through its antenna while it's wearing this electric backpack that is going to power the signals that go into its brain, and then you can use some sort of algorithm to interpret something happening in your brain into instructions for the legs of this cockroach so that it can travel along this path. That is just absolutely insane to me. So let's go ahead and dive in a little bit and see how something like this works. So let's go ahead and look back at this figure right here. So this is the setup for what they're referring to as the BTBS, which I probably would feel just fine using the word BCI for the same thing, but I'm just going to go ahead and I'm going to try to use the word BTBS for this paper because that's the word they tend to use. But BCI, CBI, in this case, even you could say BBI, so brain-to-brain interface. All of those things are pretty similar terms. So what you have here on the bottom left is just a representation of the individual wearing this EEG cap, and you can see that they're looking at a monitor, and this monitor is going to have something to do with the way that they're going to be sending signals to the cockroach. So I'm not sure if they're in the same room. Actually, I think they are in a different room than the cockroach and the S track, but they're able to see the cockroach moving along that path on this monitor. And then also there's going to be, you can see these gray squares here. So those gray squares are actually going to be really important for the way that the human being sends signals to the cockroach to tell it whether there's going to be 3 different instructions that the human can send. So you might think that the human needs to be able to send a signal to make the cockroach go forward, but actually that's not what they did in this experiment. The 3 signals that they had to send to the cockroach were stop moving. So basically the way they describe it is the rest control or the rest command, which basically is just to say it's an instruction that if it's sent to the cockroach should not continue moving. But or actually, sorry, let me rephrase that. It's not to make the cockroach continue moving. It's a sort of signal to make sure that no other signals should be sent to the cockroach. So I think actually while they're sending that rest command, the cockroach is capable of moving, but they're not actually receiving instructions to move from the human being. So, and there's some nuance to that I'll get into in a little bit. But when the human looks at one of these 2 boxes, which have a specific kind of stimuli in them, the cockroach will either turn to the left or turn to the right. So even though the human being isn't instructing the cockroach to move forward, while the cockroach naturally moves, as the cockroach starts to move forward, they may instruct the cockroach to turn to the right or turn to the left. Just one second. Okay. And then I don't think I need to explain much of this because basically what this is saying is there's Bluetooth devices that are recording information and transmitting information from the human and sending it to the cockroach. And then you're obviously getting a visual feedback to the human so that they can see where the cockroach is. But yeah, so that's about all I want to say about that. So let's look a little bit at the introduction to this paper. All right. The movie Avatar shows the dream of direct brain-to-brain control between different individuals, which has inspired researchers to develop physical communication connections between different brains. Some pilot studies have moved a step forward toward this fantastic dream through developing a brain-to-brain system, BTBS. Um, I don't like that they use the term BTBS because I don't think it translates very well to other research. I think a more common term that you'll find is BCI or BBI. Um, well, I guess they're talking about brain-to-brain systems, but I think specifically that this should be, I think a much better term would be BBI, but I think that this paper was actually written, let's see, yeah, this paper is from 2016, and that's not too old, but sometimes when you look at older papers, you'll see terms that aren't as common in sort of more recent times. But it surprised me a bit that they use the term BTBS, but it's descriptive enough. So we'll just go ahead and understand that for this paper, BTBS is something that in other papers we probably would see as BBI. So Paes Vieira developed a BTBS system between two rats based on invasive technologies, neural ensemble recordings, and intracortical microstimulation, where the decoder rat could follow the same sensory motor task as the encoder rat with the system. Yu et al. Established functional interface between brains of human and rat based on non-invasive technologies, electroencephalography, which is EEG, and focused ultrasound, where the rat's tail could be controlled by a human's intention. So basically, what they're trying to establish in this paragraph is that there is some reasonable evidence from other research projects that sort of provides the background for why they chose to do the experiment that they did. And in some research papers, the background feels more clear. But if I'm being honest, I think that there are definitely research papers where the sort of structure of the structure that's imposed by the sort of long-standing tradition of how to write these research papers can make these things feel a little bit redundant. So as I'm not going to read through all of this, but what you'll find if you do choose to read through all of this is that you might hear a lot of repetition of things or things that sound very similar. But basically, I think the message to get from this is that it's usually found in the last sentence, but the message to get from this is that we have some evidence that you can control certain physiological aspects of an animal through human intention connected to a BTBS or a BCI or a BBI. So let's see. And they're just saying that they used EEG and FUS and then TMS, which is transcranial magnetic stimulation, to do those sorts of things. But these last two sentences say, the previous studies are amazing, but the function of such BTBS is very limited. Only a simple motion or action can be accomplished, like the rat's tail being moved. This current work is what they're referring to. This work aims to develop a versatile, multifunctional BTBS between humans and cockroaches. So they're just going to try and do similar things to what have been done in the past, but they're going to try and do them in a more complex way, which is going to be controlling locomotion as opposed to controlling movement of an unconscious rat's tail. Okay, so let's see if we want to read this. A typical BTBS should consist of two key parts. I'm not going to get too into that because basically what they're going to get at is that when you have a computer interacting with a brain, you need to be able to record information and you need to be able to pass information from the computer into the brain in some way. And so brains obviously are very similar to computers in that both of them process information using electricity. But the fascinating thing about brains is that they weren't designed by someone like Steve Jobs or Bill Gates or or Alan Turing. Computers have been designed by humans. And so from the very beginning, computers had a sort of logical code that humans could understand. And we couldn't build better computers until we had a more sophisticated understanding of the information that gets passed around inside them. So the first computers were only able to do very simple things like add two numbers together or multiply, and then to do some basic calculus and do things like that. So the computers that humans have built are very good at very explicit tasks. And you can think about this in terms of how a robot might act different than a human does. A human is capable of doing really versatile things. And so that incredible versatility comes with some incredible computational load. So because of the fact that our brains are the product of evolution, we don't know the fundamental code that represents information in the brain. It seems pretty necessary that there is some sort of code like that, but it might be an entirely different thing in terms of structure as compared to the code that goes into a computer. Maybe it's not something that can be read procedurally, like from top to bottom. Maybe it's something that has to be processed all at once. And it's facts like that that make neuroscience so fascinating to me, because we're trying to do something so similar to what computers do, but we're trying to understand the way that nature has solved this same problem, and even gone beyond the sort of problems, even gone beyond the sort of problem-solving capacity that current computers have. And just a quick side note, I think that maybe the frontier that will lead us closest to understanding this level of computation will be the merger of quantum computing and neuroscience. I don't know exactly how those things will end up fitting together, but I have a feeling that the sort of computational abilities of quantum computing will be incredibly important to understanding the way that the brain processes information. So, moving on. I'm not going to read too much about these next few paragraphs just because I think that they are a little bit redundant. But one of the important things that I do want to get at is this concept of an SSVEP. So, let's see if we can find the first instance of that term in this paper. Okay, here. So, like I said before, acronyms are going to be represented by parentheses that come after a term. So, SSVEP is standing for Steady State Visual Evoked Potential. So let's just go ahead and break down what that means. A steady state visually evoked potential is a response that happens in the brain to a visual stimuli. So this is used as a way of producing some sort of very specific, very noticeable action in the electrical components of the brain. So in this particular experiment, what they're going to be doing is they're going to be showing a sort of flashing stimulus, and that flashing stimulus is going to produce a very, very specific response that can be measured through EEG. And so let's go ahead and look back down at this figure 1. So these gray boxes that I mentioned earlier are going to be the location of those steady state visually evoked potentials. And the way that each of them is going to be different than, or the way that each of them is going to produce that response in the human is genuinely by having the human look at that specific box. So let's see if we can show you a better figure. Okay, here we go. So this figure 2D, when you see these white boxes here, those are just a representation of what the human subject will be seeing as they're going through the experiment. So the 12.5 hertz box is going to be a flashing light at 12.5 hertz. And hertz is just a measure of frequency, so it has to do with how fast that flashing will occur. And so the 12.5 hertz will be in this box, 6.818 hertz will be in this box, and 8.33 hertz will be in this box. And one of the things that they mention towards the end of this paper is that the human being, it's kind of a difficult thing to just stare at a flashing light for long periods of time. And so I want to bring up, before we even get there, that even though this is one way of producing a very noticeable response in the brain, I don't think that this is a very translatable way of producing brain-computer interfaces, because who in their right mind wants to spend any amount of time longer than maybe a minute staring at a flashing light in order to do anything? I wouldn't want to do that if that was what it took to control a cockroach. I mean, maybe I'd try that once or twice, but it'd probably give me a headache really quickly. So, and not to mention the fact that if you want to look at where the cockroach is going, you have to look away from that box to look at the cockroach. And maybe you can use your peripheral vision, but even at that, it's not, I don't think it's the best control mechanism that's out there. And it's important to recognize that when we're studying these idealized situations, what we're trying to do is we're trying to create a proof of concept. And science works in this very, very slow fashion, where once you get a proof of concept for something, like using an SSVEP to do this technology, to produce this response, and to create this technology, then you look for something a little bit more sophisticated, and you want to take as small of a baby step as you can, time after time after time, and hopefully in time you can have a very solid proof that what you're doing works. Because if you introduce all of the plethora of variables that really do affect this situation, then it's really difficult to get at what's actually happening. So I understand that it's not the perfect control mechanism, but it is an idealized situation. So I just want to say these are some really funny and cool figures that I absolutely love, because most of the time in scientific papers it's just gray or black and white graphs, and this made me really happy to see. So I think that's all I need to say from the introduction. Yeah, so let's go ahead and get into some of the ways that this technology is actually set up. So I already explained basically how the human is staring at a monitor. This monitor basically looks like this right here. The human's wearing a headset that is recording from the visual cortex. So the visual cortex is in the back part of the brain. The way that you'd refer to that in scientific terms is the posterior part of the brain. The front's referred to as the anterior. So, and there's only 4 probes, and I'm going to refer to those as channels. And I believe that's the correct way to refer to them. But you might hear probes, you might hear electrodes, but these are surface electrodes. But there's a lot of interchangeable terms for these. But just know that the only recording equipment on the human in this case is a headset that doesn't penetrate the skin. It just sits on top of the skull and on top of the scalp. Okay, and then also the way that we're getting stimulation into the cockroach. So I thought this was really fascinating and honestly a little bit difficult to properly understand for me. And I think it's based on some more sophisticated previous research. And I just want to say, I am not an entomologist. I am not the best at understanding the anatomy or the brains of cockroaches, but I do know that the way that they have set this technology up is by placing one electrode in one of the antenna of the cockroach's brain, one of the antenna that sits on the cockroach's head, by sort of and I don't want to use the word skinning, but shaving down the exterior of the antenna and then placing a very small electrode into that antenna. And the only other electrode that's in the cockroach is an electrode that goes into the thorax of the cockroach, so like the body section, and that's just a grounding electrode to get a novel electrical signal. So that's all they're using to stimulate this, which I thought was really remarkable. And I don't fully understand how that works, but I'm not going to dive into that or explain that because if I'm being honest, I couldn't tell you how the motor system of a cockroach works. But it is something that can be manipulated through electrical stimulation. So let's go ahead and move on to some of the more exciting stuff. So, okay, this is pretty sciencey looking. So here we have some quantitative data, so numbers, and this is EEG data. So EEG is an electroencephalogram, sorry, but basically it's just electrical information that's almost always recorded on top of some surface, or it's always recorded on top of some surface. You can have an EEG done on the surface of the brain, and you'd have to obviously cut a person open and do surgery to do that. But that's not super typical. That's actually really uncommon. But in this instance, it's literally just a headset that this guy is putting on and putting on the back of his head. And you can see these 4 dots here. So these 4 dots represent the actual recording electrodes, the probes. And I'll get into what these. Well, actually, I think this actually might be the simplest one to explain. So these 3 rows are all EEG data, but it's EEG data that has been analyzed in 3 separate ways. So you can see that the title of this figure is Plots of EEG Signals in the Time Domain, Frequency Domain, and Spatial Domain. So I think the simplest to explain are the spatial domain and the time domain. The frequency domain is a little bit more difficult to understand unless you know about something called a Fourier transform, which I'll try and explain a little bit in a minute, but if you don't understand what a Fourier transform is, that's okay. I'll maybe put a link in the description for some good videos on learning about Fourier transforms. But just looking at this bottom row, this is EEG signals broken down by spatial analysis. So they're using they're comparing the amplitudes of the EEG signal that are occurring at these 4 different electrodes. And by doing that, they are aggregating all of the information over time, and they are basically showing where the most activity in the brain is occurring as it's recorded from these four electrodes or 4 probes. And obviously, since they're only recording from the visual cortex, you're not going to see much activity occurring at the front end of the brain or the sides, because even if there was activity there, they're not recording it. But before I go on, I should probably explain the red is going to represent higher amounts of activity, higher amplitudes, and the blue is going to represent less activity, and the green yellow is going to be sort of normal to moderate activity. And we really only need to focus on this back part of the brain because that's where all of this recording is happening. But the one difference that you can see between these different spatial representations is that you can see that over here we have some more activity on the right hemisphere of the visual cortex, and the same here. And this one even a little bit, you can see that very dark red area is a little bit to the right. I don't know exactly why that is, but I have a feeling that it could be related to eye predominance. So I'm not sure if y'all have ever heard of this, but we actually have a dominant eye in a similar way that we have a dominant hand. So uh, uh, there's a couple of different ways that you can figure out which eye is your dominant eye, but it tends to work something along the lines of you have one eye that is looking directly at things, and then you have another eye that is sort of slightly off center so that you can, and it's looking in a very, I guess it's like you have a straight line trajectory of your eye to whatever you're looking at, and you have an angled eye. So you have one that's looking, I think we often think of our eyes as both being angled inwards, but there is one eye that is more, the path between it and what you're looking at is more perpendicular to the plane of your face, and the other eye is there to provide dimension. So obviously, it would be a bit strange if both of your eyes were looking exactly forward, but I mean, maybe it's not, but you wouldn't be looking, both of your eyes wouldn't be looking at the same thing if that was the case. But if your right eye is angled inward, then that means that your left eye is the dominant eye, and your right eye is the one providing that sort of extra dimension so that you can see depth. If you only had one eye, if you can maybe experience this by closing one eye and trying to play catch with someone, you'll get hit in the face really quickly. But so each of these are, the only main difference I see is that we're getting a little bit more activity on the right side of the visual cortex. So we're probably having a lot of left eye predominance because the left eye feeds information to the right side of the visual cortex, and the right eye feeds information to the left side of the visual cortex. Okay, so before I continue, because I don't think these spatial domain EEG analyses are super significant, but what I can use them to explain is the fact that each of these columns are now representing a response to the three different SSVEPs. So if you remember what those SSVEPs were, they were those flashing lights that the human looks at in order to create a response that will get picked up by the computer, and then the computer will send a signal to the cockroach. So here we're looking, in this column, we're looking at the 6.818 hertz frequency. In this column, we're looking at the 8.33 hertz frequency. And in this column, we're looking at the 12.5 hertz frequency. I didn't mention this before, but the 12.5 hertz is the resting, is the stimulus for the resting command. And then these two are the turning commands. And I don't remember which one's which, but it's not super important. So this is the frequency analysis of the EEG signal, and this is the time analysis of the EEG signal. So one of the things that I was a little bit unhappy with this paper was that I felt like some of their figures were a little bit unclear. Like, I felt like they could have done a better job by having more obvious legends in this like for each of their figures, because they didn't specify what this blue line is, what this red line is, what this light blue, the dark blue, and all of these lines are a bit confusing, and I didn't even get a clear understanding of what the blue versus the red was here. I wanted to say it was significant differences, but again, I'm not sure about that. I think that in this, it could be broken down by alpha, beta, gamma, and theta waves, but that doesn't even sound right to me. So as we're going across the x-axis, which is the independent variable, we're looking at time in the first row. And we're looking at, on the y-axis in all three of these, and actually in this row as well, we're looking at the strength of the signal that is being recorded by the EEG. And I want to say that the reason why they're doing this is to show, I mean, the most significant one is this, because this is what's actually going to affect the experiment and actually affect the control mechanism. But I want to say that the reason why they're showing these time analyses of the EEG is just to show that there isn't some massive change in the amplitude of the EEG signal as they're looking at the SSVEP. So because you could think about it, if the SSVEP didn't produce a response that could last for 40 seconds, then if for some reason it had produced a quick EEG signal that was detectable and then sort of tapered off after maybe 15 seconds, then you would know that you couldn't have an individual trying to control a cockroach with this, because if they tried to send a signal to say, hey, turn left, and they needed them to turn left for longer than 10 seconds, if it was that sort of deteriorating signal, then after about 10 seconds, the signal would stop being sent, even though the person didn't change anything. So I think that the reason why they're showing these time analyses is just for the fact that they want to make sure that the evoked response, the evoked potential from the visual stimulus, is going to produce a steady response. And I think that's what the steady state part of SSVEP refers to. But that could be some sort of more neurologically based term. Okay, so let's read a little bit of this, but if it gets too technical, we'll back out. Implementation of Cyborg cockroach. I can read, I promise. The Madagascar hissing cockroach, or Gromphoderina portentosa, I pronounced that exactly perfectly, was selected as the model insect for the reason that it was strong and robust. Moreover, it has relatively larger size and slower walking speed. These cockroaches were purchased from Chinese market. To develop a cyborg cockroach, a microstimulator was used. 3 silver wires were used as electrodes to transfer the stimulation pulses. Okay, I have to apologize to you guys. I told you that there was only 1 electrode in the antenna and 1 in the body, but I think they said that there's 3 electrodes because of the fact that there's 1 in each antenna. So, apologies. Strands of 60 millimeter wires were taken, and the insulation was removed at either ends. One end of the electrode was soldered. I'm not going to read too much of the technical stuff. For effective insertion and minimally disturb the insects, anesthetization was applied to the cockroach through cold treatment and ice for about 10 minutes. The male connector was firstly placed on the cockroach pronotum with superglue after drying the cockroach, and then placed the ground electrode in the first segment of the thorax through an incised hole. Superglue was used to seal off all above points after successful insertion. Finally, the microstimulator could be fixed on the back of the cockroach by plugging it in the male connector, and the living cyborg could be seen in Figure 2b. So Figure 2b is this really cute little picture of a cockroach with an electric backpack. It is a little bit sad that he's got all this super glue on him, but it's all in the name of science. So hopefully one day we can super glue an electric backpack to your back. Unfortunately, you don't have antennas, so we're going to have to drill holes in your head. But it's pretty amazing to me that this is something that we can do at all. I mean, obviously, I don't think that this is something that you should just play around with. Like, I don't think that you should go out killing a bunch of bugs just for fun. But it's amazing to me that we have genuine professional scientists working on building cyborgs. I think that some of the giants on whose shoulders we stand on would be really proud of fun sentences like the microstimulator could be fixed on the back of the cockroach by plugging it in, and a living cyborg could be seen in Figure 2b. Science papers are not always this exciting. And speaking of exciting science papers, algorithms for BCI classification. So, we're not going to do this. Unfortunately, I struggle reading stuff like this, and it is important to try and dive into this material, but it's also important to recognize your limitations and your purposes. As you go through reading scientific material, you want to make sure that you're not just focusing on information that is only germane to the projects that you want to do. You also have to be open to the idea that there is information that is counter to your projects, and not just reject that because you want to have an easy project. But when it comes to information that is too dense and genuinely just makes you want to bang your head against the wall, it is okay to skip over it sometimes. So I would love if somebody could explain some of this stuff to me. I don't have the math background yet to truly understand what's going on here, but I would really like to. And but to be honest, the purpose of this video is more so to focus on the procedures and then some of the very fundamental neuroscience concepts that go into a project like this. So I am going to skip over the algorithms for BCI classification section, but I want to mention that basically half of this research paper is focused on exactly this algorithm. So, like they said in the introduction, there have been other examples of modulating the behavior of some sort of animal or insect uh, through these BTBS systems or these BCI, BBI systems. But one of the important things to do as we progress in research is to try and create more accurate systems and to try and reduce the amounts of error that exist in these systems. And that's exactly what this classification algorithm is for. Basically, they're going to be looking at the EEG signals coming from the human brain in response to these SSVEPs, which are the visual stimuli. And the classification algorithm is responsible for trying to make sure that there is only a valid detection of intention in the human brain when the human actually intends to make the cockroach move. And there was already a pretty high level of accuracy with this specific system, but you have to understand that we're using a very kind of crappy stimuli. We're using SSVEPs. We're making a person stare at a flashing light. So that is a very controlled system. So even though they're trying to maximize efficiency with this very controlled system, this classification algorithm may be one of the most important things that comes out of this research as it gets applied to more and more sophisticated types of control mechanisms. Like, I know that one of the other types of control mechanisms is the idea of a human being imagining raising their arm. And that is not going to produce as explicit of a response as maybe staring at a flashing light. So it's important to make progress in the sort of systems that help make computer-brain interface more accurate, even if we're doing it in a very idealized situation. So I'm not going to go too much into the algorithm because I don't understand it perfectly well, but I do want to say that it is an important part of what they're doing in this paper. So let's scroll down through all this beautiful, beautiful work. And then here's a human subjects and insects and ethics statement. So basically they're just giving information about the gender of the subjects, and they had all males, and that's probably just for uh, trying not to introduce other variables. Whenever you're doing science, it's best to try and cut down on variables that aren't actually a part of the scientific experiment. If they were interested in the ways that female brains versus male brains produce these responses, then that would be a very valid variable to manipulate. But it might be more valuable to control your population as tightly as you can. And it doesn't have to be males, it could be females. But in this instance, they used males. And then they describe the cockroaches a bit. And here they go into the experimental protocol, which I'll explain briefly, but I think that you probably understand the way that it's working. And really, the main reason why scientific papers add in or have a section on experimental protocol has to do with allowing for their experiments to be repeatable. It's important to lay out your experiment in a way that if somebody else said, bullshit, I don't think that you really did what you did, and I don't think those numbers make any sense. It's important for you to say, okay, well, if you think that what I did is bullshit, then go out and do it yourself. Here, I've written down exactly what you have to do, and you don't even have to figure out how to do any of these things other than to just follow my step-by-step instructions. So a diagram like this is really just to lay out very explicitly the way that they gathered data. So they had two phases. Phase one was there's a training phase within these things, but phase one itself was sort of a calibration phase. So in that section, as opposed to having an individual actually try and control a cockroach, what they had was they would have a rest period, and they would have a period where they would be told, okay, look at this box. look at this box, look at this box. And as they were instructed to look at those boxes, the prediction algorithms were being tested with the optimization algorithm and without the optimization algorithm. So the prediction algorithm is or I don't want, maybe prediction algorithm isn't the right word I should use. They were trying to classify the intentions of the human by looking at EEG signals. And if you're trying to use intention to control a cockroach, then it's difficult to tell whether or not your optimization algorithm is helping. Like, you definitely want to try and get better results, quicker times through the S maze, and more accurate turning signals, which you could record in some way by asking the person, "Hey, did you mean to make the cockroach turn now?" But that's not a very stringent experiment. So in phase 1, what they did was they had the individual have specific times where they would look at the left turn stimulus, or look at the rest stimulus, or look at the right turn stimulus. And because they knew at what times the person should be looking at those things, they could also use their classification algorithm and use it with or without their optimization added into the algorithm, and see, did they actually predict the which stimulus the person was looking at in a way that corresponded with the times that they were instructed to look at those things. So I'll try and clarify that and say that one more time. When they were told to look at the left turn signal, they had, let's say, 40 seconds to do so. If their prediction algorithm, their classification algorithm, predicted, just using its own methods, what they were looking at correctly, then that was like a good thing. But if it didn't correctly predict left turn, then it counted against that prediction algorithm. And they wanted to compare the accuracy of the prediction algorithm, the classification algorithm, with and without optimization added to it. So I know we didn't talk too much about the optimization algorithm, but basically phase 1 was all about trying to see whether or not their optimization algorithm helped to make the classification system more accurate or not. And then phase 2 is the main experiment that we've been talking, or I don't want to say main, but it's the experiment that we've been talking about the most, navigating a cockroach through that maze. So this is basically saying that they broke up the trials in a specific way, which isn't super important. It's basically just to say that they tried to be careful that they didn't have confounding variables in the experiment. And that's pretty much all it said in here. So results. Let's look at some results. I'm not going to read too much through this, but I will tell you all some terms that are important to understanding this figure, because the figures are almost always the thing that is going to take up the most time and the most attention and give you the most information. So and this is called the figure legend here. So the figure legend for figure 5 says, experiment results before optimization and after optimization. So BO, not body odor, is going to be before optimization, and AO is going to be after optimization. So it's got 3 parts to it. It's got part A, B, and C. So A is going to be looking at classification accuracy. So classification accuracy refers to how accurately the computer is able to recognize the intentions of the human being. So all of this information is about how accurately the computer was able to pick up the intentions of the human. And then on to B. This is the online classification accuracy for all subclasses. So the subclasses just refer to the different frequencies that represented the different stimuli that correlate to the different intentions, like left turn, right turn, or rest. And then C is basically the same thing, but it's including some of the periods of time where the individual was not looking at any of the specific boxes that had the stimuli in them. It includes the time in which the human was looking between them. So it's a little bit more like real life in that sense. Or it's a little bit like it's less carefully chosen to make sure that the results are significant, which is an important thing to do. But I think it's also good that they separated these things out. So, and actually, one last thing I want to say is when you see this p less than 0.001, that's some statistics that I'm not going to explain right now, but basically having a significant p-value, seeing those stars, usually means that they found something in their data that you should actually take notice of. You might hear me say that they found a statistically significant result, and that is exactly a statistically significant result is a result that you can be pretty sure didn't happen by chance. It was something that happened, and there's a real trend there. So I'm not going to go too much through this because there is a ton of stuff here. The main thing I'm going to focus on are the mean values, the average values of all of the trials. So here, let's look at these legends first. So I'll zoom in a bit. Okay, so we have 12.5 hertz, 6.818 hertz, 8.33 hertz, and the entire spectrum, or the entirety of all of these. And we're looking at how accurately those stimuli produced a response that the computer could pick up. And we're not seeing a statistically significant difference between them, which is fine. But the important thing to notice is that the average classification of these intentions was around 90%. That's really good. So if you think carefully about what that means that the computer is understanding the intentions of the human being at a rate of at a proportion of about 90%. So that to me, that's a pretty good number. So now let's look into these. And so the main thing it's trying to do here is just to break these things down by it's breaking them down by which stimulus is being looked at. So you've got the 12.5 hertz, the 6.81 hertz, and the 8.33 hertz. And this figure is really focused on the optimization algorithm. So BO represents before optimization and AO represents after optimization. So what you're going to want to see if the experimenter's optimization algorithm worked well is that you're going to have a statistically significant difference between the before optimization value and the after optimization value. The before optimization value should obviously be lower because you want the optimization algorithm to make your predictions or your classifications of human intention more accurate. So let's see if we found that. I think the best one to look at here is going to be the entire experiment in before optimization to after optimization. So that's going to be this mean value here is going to have the most data aggregated all together. So I think that is the one that will be the most telling of the trends. And look at that. You see those stars right there? Let's see if we can get closer. Oh man, that's kind of not fun. Let's see. Oh my gosh. Okay, that'll work. Okay, so this is before optimization and this is after optimization, and this is for the entire experiment. And we've got 3 stars right there. It's not Yelp, so you don't need 5, but 3 stars, and you can see that the after optimization is a lot higher than the before optimization. And if you're wondering what these bars are here, basically it's a really, really good thing to get a difference where the difference you're looking at shows that one of these bars is out, or one of these colored bars is outside of that sort of weird stumpy leg thing. And for you people that know statistics, you're going to think that I'm really dumb for explaining it this way, but statistics is really scary to a lot of people. So just know that if you see one bar that is out of the reach of this stumpy leg thing, that's a very big difference. Okay, so moving on. Let's just quickly say the big difference there shows us that the optimization algorithm seems to really help with the accuracy of these predictions. Like that blue bar that we were looking at right there. Let's see if I can get it back. Yeah, so this blue bar right here is at about, let's see if I can get it. It's at about, that's not going to be good. That blue bar right there is at about, oh goodness. Let me make sure, ooh, the dependent variable there is time. Oh, that's the recognition time. Sorry, let's go over to, my bad, y'all. It's important to admit when you're wrong, and I was wrong there. But fortunately, the same trend holds true when we're talking about accuracy. Because I thought I was talking about accuracy, which is actually showing up in part B. But so this is basically saying that the optimization algorithm reduces or increases the response time in when you use it. So basically, it means that the optimization algorithm takes longer to send the signal to the cockroach. But what I was trying to say is still true in part B, which is that the after optimization has a statistically significantly greater accuracy value than the before optimization testing does. And you can tell that from the 3 stars right there, and the fact that the green bar, which is entire BO, is lower than the entire AO. So sorry about that, but who cares? Okay, so all right, this is maybe one of the cutest figures I think I've ever seen in any experiment ever. So let's just take a second and enjoy the fact that in this experiment, they really got to drive cockroaches. And that's exactly what's going on here. This is just time-lapsed photos where they're showing the path. This cockroach went in a little circle right here, and then he went over there, and they let him walk forward, and then he made him make a little right turn, and then he took a little left turn. And I just think it's so cool that you can actually do stuff like this. Okay, so this graph was absolutely horrible in my opinion. I really didn't like it at all. They overlaid so much information, and they put metrics on one side and on the other side at the same time, which I don't like. And you see how many different metrics are all going on at the same time. I don't like the way that they did this figure, and it's pretty well summed up in the table that goes beneath it. But basically, they're just providing the numbers for the accuracy of the classification of these things. And I'm not going to go too much into that because I don't think it's super important for our purposes, except for the few things that they will mention in the discussion, which is the last section. So I'm going to go ahead and go to that because I think that'll provide the real juicy meat of this paper. And often that's the way that scientific papers read, is that most of the paper is pretty much directed at someone that wants to verify the results of this paper. And if you're reading a research paper, most of the time it's not really your responsibility to verify the results. Although you want to be careful that you're not looking at results that are very blatantly sort of false, or there's some sort of serious confounding variable in the experimental procedure. But unless you see something like that, I don't think that you necessarily have to get too deeply into the nitty gritty. The main thing that you're going to want to look at is going to be the discussion, because that's where the scientist is allowed to have a little bit of free reign and say, oh, well, what did you actually think was going on, and how do you explain the results of your experiment? So I am going to read through this whole discussion. It's not too long, but I think that this is maybe the most exciting part of the whole paper. Okay. The present study demonstrated that humans' intention, extracted from the subject's EEG, could be transmitted to partially dominate an insect's brain. The human could successfully steer a cockroach to make turns using thoughts through SSVEP-based BCI and microinvasive neural stimulation. The SSVEP could achieve a high CA, which is classification accuracy, so that has to do with recording the intentions of the human brain. So if you see CA, it's classification accuracy. The SSVEP could achieve a high CA of 90% by only using 4 channels on the scalp of the visual cortex with a portable and wireless EEG device in the training phase. I think that's a really important point. The device that they used was portable and it was small and it didn't require any surgery to be done on a human. So the fact that we're showing that control systems using very reasonable and efficient technology can be made, and they can be used to do incredible things like control a cockroach. So we also proposed an optimization algorithm for SSVEP, which greatly eliminated the random errors which occurred during online control processes. As a result, it significantly increased online classification accuracy by 8%, from 83.34% to 91.42%, with the removal of frequency transient state signals. So we'll go on before I get into that, because I don't want to confuse things as we're going. Generally, misclassification appeared during shifting period of a subject's gaze in SSVEP. Therefore, this phenomenon might be improved by adding a program to detect transition of the EEG signal on frequency before classification and further study. Even though such increase of online classification accuracy was possible at the sacrifice of SSVEP program's fast response performance. So basically they're saying that the optimization algorithm increased the amount of time that was required in order to send a signal to the cockroach. Besides, regarding the present SSVEP-based BCI, due to high brightness and repetitive stimulation, the user might suffer fatigue when using SSVEP for a long time, where fatigue was generally a feeling of tiredness, reduced concentration when performing a task, or reduced concentration when performing a task. The amplitude and SNR of the EEG signals were consequently affected by the fatigue. One possible solution to reduce fatigue of SSVEP was to use visual stimulation with high frequency or modulated amplitude. By implementing the possible improvements on SSVEP in further research, we can move current systems a step closer to practical applications. So I think it's important they recognize what they did in that paragraph, because they're saying that they understand that it's not super reasonable to expect a person to stare at flashing lights in order to control anything of real application. And they're saying that maybe there are some ways that these experiments could have less of a fatiguing effect on the participants just by doing things like increasing the frequencies, so using higher frequencies, which personally kind of to me seems like it would increase fatigue, but there's probably research behind what they're saying. And then also varying the amplitude, so having some brighter and some not as bright. That could just make it less monotonous over and over, but it's still pretty monotonous. The other similar studies were compared with our work as well. Pais, Vieira et al. Succeeded in building up a BTBS for real-time sharing of sensory motor information between 2 rats with intracortical microstimulation, in which the decoder translated correctly 64.32% the motor information from the encoder under a time delay of 20.06 seconds and 13.59 seconds. So they're saying that this other group that has done something similar in rats with tactile information, they had a much longer time to record what was happening in their brain and then send the signal to another. So the encoding took 20 seconds and the decoding took almost 14 seconds, whereas in this experiment the encoding and decoding was happening around 1 to 2 seconds. Another experiment in the research demonstrated the decoder translated correctly encoders 62.34% tactile information, and the time delay was 2.66 seconds and 2.68 seconds respectively. Recently, Rao et al. Accomplished a direct BTBS between humans, where the motor imaginary, I think that they might mean imagery. I think that they probably do mean imagery there. So I don't like seeing typos and stuff in, especially in important words within research papers. But it's besides the point at this point. And TMS were applied to complete a task. The paired true positive rate varied from 83.33% to 25%, and paired false positive rate varied from 0% to 37% in online experiments, with a 2-second setup time plus a 20-second visual countdown in each trial, and a transmission duration of about 650 milliseconds along the system. And now they're going to refer to their own experiment. In this work, cyborgs, the receivers, produced quick and accurate responses with a response accuracy of about 90%, left turn accuracy of about 84%, and right turn accuracy of about 92%. Through applying the invasive technique of neuromodulation, where monopole square pulse trains were indirectly sent to the brains of the cyborg cockroach via antenna. And when they say monopole square pulse trains, that just refers to the fact that they were sending a signal that was on entirely and then off entirely, instead of something that gradually increased and then decreased again. And the time that a command sent from SSVEP to the completion of reaction from the cyborg was about 772 milliseconds. In addition, the techniques we used in the BCI segment in this study showed high decoding accuracy, which was about 86% classification accuracy of the sender's intention, as well, and the average decoding time was 1.79 seconds. So basically, you can see that they are getting really, really quick classification times and response times. So when the human is sending this signal to the rat, it's taking almost 2 seconds, but under 2 seconds, to get the information out of the human's brain, and so the computer knows what to send, and then another about 0.7 seconds to actually get the response from the cockroach. So even though 3 seconds may seem like a long time to you, how long do you usually take to use mind control on a cockroach? All right, going on. In the work of Latif et al., they tried to steer cyborg cockroach to walk along an S-shaped lap in 2 directions manually, and succeeded in achieving a success rate of around 10%. So when they say that they were doing it manually, as opposed to decoding human intentions and then controlling the cockroach, they were just controlling the cockroach with computer commands. However, a successful online navigation of Cyborg with human brain in this study was much more challenging, which required stable and continuous high-level accuracy in both sender and receiver sides. The experimental results demonstrated the BCI could successfully control the cyborg to walk along the S-shaped track in all 3 groups. And the success rate ranged from 0 to 50% in 10 trial experiments, where the mean success rate was 20%. The value was not high enough, but significantly above the level in control group, which was 0%. Which basically means that when they weren't controlling the cockroach's motion, none of them completed the S track. I think that's what they're saying there. But besides, I think that we'll get the point even if we move on. And the SR, that's the, let me check, see what that is. Do y'all know what it is? The success rate. So, and the SR coincided with the roughly estimated value based on the accuracy from both sender and receiver parts, as there were 12.7 turning commands in average during each online trial. Additionally, the CPC, which is just a coefficient of whether or not they were accurately classifying and sending correct signals. Additionally, the CPC as an indicator of system performance reached 0.616, while its chance level was 0.375. Above all, the results achieved through the system in present experiments were rather optimistic and instructive. In addition, the architecture of current system still had the potential of enabling developing multi-mode control cyborgs, more than 3 modes. And those 3 modes refer to, I believe they refer to the rest command, the left turn command, and the right turn command. And increasing the control modes might contribute to the improvement of success rate in online control tasks, and at the same time enable more precise online assignments as well, which would be designed and validated in further study. So basically they're saying that they think maybe the success rates in these sort of control mechanisms could be greater if they could do more things besides just command them to rest, turn left, and go to the right. It is meaningful to build up a BTBS between the human and the animal. Firstly, this all-chain wireless system proves that it's possible to build up an embryonic virtual brain-to-brain information transfer device with current technology. Besides, the idea also has the applicable value in reality. For example, it could be further used for detection in complex and dangerous environments. Most importantly, as a pioneer study of brain communication between human and animal, the successful implementation of BTBS expands the possible usage of both BCI and neuromodulation. Furthermore, the alternative BCI and neuromodulation modalities, both invasive and non-invasive, may have huge potential of intersection and integration to create far-reaching usage in future. We have seen a mushroom growth of internet based on computers during recent years, and perhaps brain-to-brain network may be developed in the future, just as the science fiction films predict. They actually reference science fiction films in the last sentence of this research paper, which I think is so fantastic. I mean, science can be, as you may have felt during this video, incredibly boring, but science fiction is, I would say, about 90% of why anyone does science. I think that we all sort of have this dream in our heads of, I don't know, making humans live forever, or giving humans wings, or like me wanting to be able to control your mind so that I can put you on a beach and put a margarita in your hand, and then you can drink it and enjoy it and get drunk even, and then as soon as you want to turn it off, you can turn it off. I think that would be amazing. And there's so many more things that I want to do with brain-computer interface. Specifically, my interests are in brain-computer interface as it relates to language. I think the idea that one day, as opposed to reading out whether or not you're looking at a flashing light that's going at 12 hertz or 6 hertz, I'd like to be able to read information out of your brain, such as what words are you thinking of? What story are you thinking of? Could I even have you think of a movie and pull out the entire movie from your brain and put it into someone else's brain, and you can both watch a movie that one of you is writing in real time? And all of those things may be unrealistic in some sense, but I don't think that they're too far gone from what is actually possible. And I think that if you're not really privy to neuroscience, then watching a video like this, you may have thought that mind controlling a cockroach was something that wouldn't happen in a million years. And honestly, sometimes I feel like I'm a bit out of the loop on what is normal and what's not normal, because to me, when you say, yeah, you can control a cockroach with your brain, and just by looking at different things, you can tell it to turn left or right. For me, that seems perfectly reasonable. And I think that there's probably a lot of people out there that hear the idea of mind control and steering a cockroach with your brain and just instantly think that it's never going to happen, and that it's a totally crazy thing to think about anyways. And so I'm so glad to see a science paper written in a way that sort of allows for a person to think about the amazing things that science really could do. And I hope that you do that too. I hope that if you are watching this video, that this inspires you in some way. Is there something that you think is impossible to do that maybe you could do a PhD on? I mean, there will always be things that are impossible, but how close to those things could we get if we really tried? And I think one of the best ways to get to the impossible is through science. So I hope you enjoyed this, and I hope that I didn't fumble around too much, but I'll see you next time.